everyone, this is LATHA AKULA. From today onwards, we are starting the new lesson, that is biomolecules. So, if you see on our earth, there are a wide range of living organisms are present, right? So, almost all living organisms, they have the same chemical composition. Do you believe it or not? Almost all living organisms, they have the same type of chemical compounds present in it. For example, if we take a plant tissue or animal tissue or a microbial culture means if we grow the microorganisms in the laboratory in the petri dishes, we'll get some microorganisms, that is called as microbial culture. And all these consists of same type of chemical elements. They were carbon, hydrogen and oxygen. And if we take the non-living elements like rock or earth crust, they also consist of the similar type of elements. If we deeply analyze the chemical composition of these two, we'll find the carbon and hydrogen concentrations much higher in the living organisms when compared to the non-living matter. And for identifying the chemical composition, what we need to do, we have to make a chemical analysis of those compounds. So for that, what we will do is, we will perform the chemical analysis by using a living tissue. What type of the living tissue? We'll take either plant tissue or animal tissue. Plant tissue means we'll take either leaf or a root or stem or a fruit or vegetable, anything which it is derived from the plant. And not only that, we also take the animal tissue also, like liver tissue. So that was taken in the mortar, and then we do this, we add trichloroacetic acid. What is this? This is the chemical substance. It is added to the living tissue. What is its function? Its main function is to lysis the cell. So, to analyze the chemical composition of the living organisms, what we have to do? We have to break the cells present inside the tissue, right? For that, we are taking the trichloroacetic acid, which will cause the cell lysis or cell breakdown. So, to the living tissue, we have added trichloroacetic acid, and both of them was grinded into a thick paste. Finally, we get a semi-solid thick paste is obtained, right? That is called a slurry. That slurry is strained. For straining, what we will do? We'll take either a test tube or a beaker. So to the beaker or a test tube, what we will keep? We'll keep a funnel. In that funnel, we'll keep either muslin cloth or filter paper or cotton. By keeping that cotton inside the filter, inside the funnel, we'll pour this slurry, and what we'll get after filtration, whatever the liquid substances or whatever the minute particles, they get filtered. And the particles are large substances which are not filtered, that get retained in the funnel. So the liquid part which is filtered are called as filtrate, and the remaining part which is not filtered, which will be retained in the funnel, that is called as a retinate. This is the filtrate and this is the retinate. And this filtrate is also called as acid soluble pool, and whereas this retinate is called as acid insoluble pool. So we need to remember the liquid portion of the filtrate is called as acid soluble pool, and the remaining part which is retained in the funnel is called as acid insoluble pool. If we perform the chemical analysis of these, what are the substances present in the acid soluble pool and what are the substances present in the acid insoluble pool? In the acid soluble pool, all organic compounds are present, whereas in acid insoluble pool, they consist of macromolecules like proteins, nucleic acids and polysaccharides are present. What are present in it? Proteins, nucleic acids. What is meant by nucleic acid? DNA and RNA. So, nucleic acids and polysaccharides. Polysaccharides are also large compounds, right? So, those are present in the acid insoluble pool. So, here this is about organic substances present in it. So, in the living organisms, not only organic compounds but also inorganic elements are also present. How to analyze the presence or absence of inorganic elements? To identify the inorganic elements present in the living tissue, we perform ash analysis. What is the test done to identify the inorganic elements present in the living organisms? Ash analysis. So for this, what we'll take is, we'll take any living tissue. After taking the living tissue, the wet weight of the tissue is weighed first. So wet weight means what? It includes water also. The living tissue consists of water. Inside the tissue there is a water is present, right? So along with water, the weight was measured, and after that, the tissue is dried. During the drying process, what will happen? Whatever the water present in the tissue that get evaporated or that get removed. After drying, after complete drying, the dry weight of the tissue is again measured. Here we have weighed along with water, that is called as wet weight, and then here we have removed the water, and after that we have measured the weight, that is called as dry weight. After measuring the dry weight, the tissue is burnt completely. So during the burning process, what will happen? We'll get some fumes from the right? So during that process, what will happen? The carbon compounds that get oxidized in the form of carbon dioxide and water vapor, and the remnant is called as ash. So after the fumes have been removed, the remaining tissue which is completely burnt is called as ash. Remnant is called as ash. So if that ash was chemical analyzed, that consists of inorganic elements. So all the inorganic elements are present in the ash. And what are the elements present in it? Calcium, magnesium, etc. So calcium and magnesium are present in the mostly found in the in the ash of the living organisms. And if we see some of the elements present in the earth crust and human body, these are the some elements like hydrogen, carbon, oxygen, nitrogen, sulfur, sodium, calcium, magnesium and silicon. If we see the percentage of these chemicals, hydrogen, it is much higher in earth crust when compared to the human body, and the percentage of carbon is 0.03%, that is more in the human body, that is 18.5%. And if we take the oxygen, the percentage of oxygen is more in human body, that is 65%, whereas in earth crust it is 46.6 only. And if we take the nitrogen, very little amount of nitrogen is present in earth crust, but whereas in human body, we have, our body is made up of so many proteins, right? That means it consists of more amount of nitrogen, that is 3.3% of protein nitrogen is present. And now sulfur is 0.03%, and whereas in human body it is more, that is 0.3%. If we take the sodium, the percentage of sodium is more in earth crust, that is 2.8%, whereas in the human body is only 0.2%. And calcium, the amount of the calcium is also more in the earth crust, that is 3.6%, whereas in human body it is only 1.5%. If you take the magnesium, it is 2.1% in the earth crust, whereas in human body it is only 0.1%. And the last one is silicon. The silicon is nearly 27.7% is present in earth crust, but that is not found. Very negligible amount of silicon is present in the human body. These are the several elements present in the human body. And now in the next we will see about what are the inorganic elements present in the human body.